You probably recognize these as all protein-rich foods, so meat and eggs and some vegetables are all very rich in protein. And proteins do more jobs in the cell than any other type of biological molecule. Many different parts and structures of cells are actually made of proteins, and enzymes that control the thousands of chemical reactions that maintain life's processes are also proteins. Proteins are made of the elements carbon, hydrogen, oxygen, and nitrogen. In addition, sulfur is often present, and sometimes phosphorus and other elements. These elements combine to form building blocks of proteins, which are called amino acids. So, amino acids are the monomers of proteins. When you link them together in a chain, they become proteins. And there's more than 100 different kinds of amino acids in cells, but only 22 join together to form proteins. The general structure of an amino acid contains a carbon molecule, right here in the center, that is bonded to an amino group on one side and a carboxyl group on the other side of that carbon molecule. Also bonded to your central carbon is something which is referred to as an R group that forms a side chain. And the R group is actually what distinguishes which amino acid it is. So if you look down here at these three different amino acids, glycine, cysteine, and tryptophan, they all have that same structure or basic structure. They have an amino group, they have a carboxyl group, both of which are bonded to a central carbon. However, the R groups, highlighted here in purple, are all quite different. This one just has a hydrogen attached to that central carbon, and these are more complex molecules. And there are 22 different amino acids, each with a different side chain that form proteins. So just like other macromolecules, amino acids can join together through dehydration reactions. And the bond that is formed between amino acids is called a peptide bond. Two amino acids linked together form a dipeptide, di for two. Three form a tripeptide, peptide equals 3 amino acids. Chains of fewer than 100, so less than 100 amino acids, are called peptides. And greater than 100 amino acids are called polypeptides. So this illustration shows two amino acids which are going to join together to form a dipeptide through a dehydration reaction. So the same for other biomacromolecules. You end up with a hydroxyl group from one of the amino acids and a hydrogen, which in the process of forming the new bond are given off as a water molecule, resulting in a new bond between the two amino acids forming your dipeptide. And the reverse reaction is, of course, hydrolysis, where you have to add water back into that reaction to break the bond and form two amino acids again. It is the order and number of amino acids that make the different types of proteins. The ordering of the amino acids and proteins are determined by the genes in our chromosomes. Plants can make their own amino acids. Humans can make most amino acids from starch, but there are 8 essential amino acids we must obtain from protein-rich foods such as fish, dairy products, or tofu. We rely on digestive enzymes to break down peptide bonds and release amino acids from proteins in the foods that we eat. And our body can then use these monomers to build its own polypeptides. Most proteins don't exist as long chains inside of cells. Instead, the peptide chain folds into unique three-dimensional structures determined by the order and kind of amino acids. The structure of a protein is described at 4 levels. The primary structure is simply the amino acid sequence of a polypeptide chain. Primary structures can then form into one of 2 different secondary structures, either an alpha helix or a beta sheet, as a result of hydrogen bonds between parts of a polypeptide. So hydrogen bonds are responsible for primary structures becoming secondary structures. Interactions between R groups, remember the R groups, which distinguish the difference between different types of amino acids? Well, those R groups can cause proteins to fold into a tertiary structure. When you get multiple tertiary structures interacting, they form quaternary structures. So for example, this protein consists of 2 polypeptides. One tertiary polypeptide up here, which is dark purple, and another lighter polypeptide down here. And when they interact together, they become a quaternary structure. Hemoglobin is an example of a quaternary structure protein. And this is a protein that carries oxygen in your blood. And it actually consists of 4 polypeptide chains. A protein's function depends on its overall shape. So this is another example of structure relating to function in biology. Proteins are therefore vulnerable to conditions that alter their shape, because therefore that protein will no longer work. So things like heat, excessive salt, or the wrong pH can disrupt the hydrogen bonds that maintain the protein's secondary and tertiary structures. This then denatures the protein. So this is called denaturation, and it results in a loss of function. The protein can no longer do its job. The protein is denatured if its structure is modified enough to destroy its function. So what happens to eggs when you cook them is a good example. The white of an egg is mostly made of protein. Before you cook the egg, this protein is clear, like the white in the center of the egg here, closer to the yolk. Once you cook it, it turns white. And this is simply because you have denatured that protein. Similarly, fish turns from translucent to opaque as it cooks. Most denatured proteins will not renature. In other words, they won't reform their protein shape that enables them to function, to do their job. There's no way to uncook an egg, for example. Gentle denaturation, however, is sometimes reversible. You will get more detail about this when we start studying enzymes.